All right, hello everyone. I'm going to go over other types of inheritance. So I'm going to do a quick video on examples of how to solve the problems in that last section of the genetics practice worksheet. The Bozeman video went into some of these things. I'm just going to go through some examples. Okay, so the first thing that we are going to do a practice problem for is sex-linked traits. I'm going to post these slides, but sex-linked traits are basically genes that are found on the sex chromosomes, which are going to be X and Y. Almost, I would say like 90% of the time, the questions will be referring to the X chromosome. All right. So with sex-linked traits, you have to take into consideration the sex chromosomes when you're writing the genotypes. So for males, typically have X and Y as their sex chromosomes. Females are XX. So when you do the genotypes, you actually have to include the X and the Y. And you write the letters, so the like big A, little a, as a superscript, like so as like an exponent on the X chromosome. So for example, this one right here is about color blindness. So color blindness, it says, color blindness in humans is a sex-linked recessive trait found on the X chromosome. A female who is a carrier for color blindness has a child with a male who has normal vision. So I'm actually just going to make a little key. Always make a key. So I'm going to do capital A is going to be normal vision. And I know that because in the problem it says that color blindness is recessive. So little a is going to be colorblind. Now, when you write it, because it's sex-linked, you have to now include the sex chromosome. So because this is our first practice problem, for a male, X, let's say capital A, Y. So this person has a normal vision allele. This male would have normal vision. Whereas if they had X little a, Y, and notice, remember, so it's like you write it like an exponent. This would mean that they are colorblind. For males, since males typically only have one X chromosome, it's either normal in this case or colorblind. They only have two options as their genotype. Now for females, there's going to be a little bit more options. So a female could be X big A, X big A, since they have two X chromosomes. This will be normal. They could have X big A, so one of their X chromosomes has normal vision allele, and then the other chromosome has the colorblind allele. They would be heterozygous, right? Because you have one capital, one lowercase. But their phenotype, so in terms of their vision, they would still have normal vision. All right, so they still have normal vision because of this dominant allele. And let me, but we would call these women carriers, or females carriers. So a carrier means that they're heterozygous, they carry the recessive allele, but their phenotype is going to be the dominant. And then the last option for a female would be X little a, X little a. In this case, since they have two recessive alleles, they would be colorblind. When we do sex-linked traits, this will be pretty much the same, like you use a similar key each time. I always put a key indicating what is the dominant allele, what's the lowercase allele. And then when you are talking about the male parent versus the female parent, as long as you include the X and Y chromosomes, and then you do the alleles as exponents, that would be how you indicate the genotype. All right, so going back to this question. It says a female is a carrier. Okay, so the female carrier is going to be this genotype. Okay, so she's a carrier. And then the male has normal vision. So the male would be X big A Y. So those two, the ones I highlighted, are going to be our parents. You set up your Punnett square. all right? Because of Mendel's laws, they segregate. Okay, so these alleles segregate. So you have X big A and Y. And just kind of as a little recap in terms of drawing, so that means half of this person's sperms carry this X chromosome, half of the sperm carry the Y. For the female right here, you have X big A, X little a. That's her genotype. Once again, each of these, through Mendel's laws, they segregate. These are individual egg cells. Fertilization. So when you match them up, X usually goes first, and then capital letters go first. All right. So this first individual offspring will be X big A, X big A. The next one right here, X big A and Y. This individual, X big A, X little a now. Okay, so this one's gonna be heterozygous. And then finally, our last one is gonna be X little a, Y. So these are possible offspring. Now, because this is sex linked, you have to divide your results based off of whether you have a daughter or a son. So the first question is, what is the chance of having a daughter who is colorblind? So cover up the boys right here or the males, XY. If we look at the females, this first female, X big A, that would be normal vision. X big A, X little a. This female also has normal vision, right? So the chance of having a daughter who is colorblind, it's going to be a 0% chance of daughter who, oops. Oh, I should have done my nails. So gross. All right. Okay. 0% chance of daughter who is colorblind. And then the second question is, what is the chance of having a son who's colorblind? Now this time cover up, ignore the females on the side. So you have 50% chance of a son who has normal vision, and then 50% chance of having a son who is colorblind. So in this case, it would be a 50% chance of son who is colorblind. All right. So in this example, if they had a daughter, XX, those females would not be colorblind. But if they had a son, there's a 50% chance that the son might be colorblind. Because in this case, since females have 2 X chromosomes, I like one of the 9th graders said, she described it as like the X chromosome. Since females have 2, it's kind of like females have a backup. Whereas males, since they only have 1 X chromosome, they either got it or they don't. All right, so in this case right here is a 50-50 chance for a male to be colorblind if you had these 2 as the parents. Okay, let's do one more example. All right, so another example is in fruit flies. Fruit flies also have X and Y chromosomes. So they would still follow the same as humans, XY for male, XX for female. In this case, so example 2, this question says, in fruit flies, eye color is a sex-linked trait found on the X chromosome. Red eyes are dominant to white eyes. So I'm going to make my key. Capital A equals red eyes. Lowercase a equals white eyes. All right, so it says a female red-eyed fly who is heterozygous mates with a white-eyed male. So once you make your key, you write down what the parent genotypes are. So the female, XX, and it says that she has red eyes, but she's heterozygous. So one is going to be capital A, one will be lowercase a. The male has white eyes. So the male, XY, as the chromosomes, since they have white eyes, this X chromosome is carrying the recessive allele, the little a. All right, so these are our 2 parents. And then if we do the Punnett square, okay, I'll pause and then fill this in. All right, so I filled in the Punnett square, and the question is asking, give the predicted phenotype proportions of their offspring. So just to kind of save time, you have female. This female is heterozygous, but their phenotype will be red eyes because it's one capital A, one lowercase a. This female has 2 recessive, so this female will have white eyes. I'm just using gray for now. This male will have red eyes, and then this last one would have white eyes. Depending on the question, though, they might be more specific. So, like if they have a female offspring fly, what % chance would it have for white eyes? Or just make sure you answer, like read the question. But this would be how you would set up the Punnett square. Okay, so this is how you would do a sex-linked trait. And once again, I would say almost 90% of the time for AP, we're talking about traits that are on the X chromosome. Okay, another type of inheritance is going to be incomplete dominance or codominance. Now, they aren't the same. Incomplete dominance and codominance aren't exactly the same, but I'm grouping them together because you would solve them in a similar way in a Punnett square. So what we've been doing thus far, we've been talking about complete dominance, meaning capital A, whatever the dominant trait is, completely covers up the recessive. Incomplete dominance, it's known as blending. So incomplete dominance is like blending, a mix of the 2 alleles. So if you look at these pictures right here, if you have a red flower and a white flower, this right here shows you that if the flower was homozygous dominant, they would be red. Homozygous recessive, it would be white. But if they were heterozygous, instead of just being red, because red was the dominant color, incomplete dominance is showing that the heterozygous individuals are this pinkish color. So this is an example of like blending. Another example is these cows. It's kind of these heterozygous cows are mixed between black and white. Or sorry, it's not black, it's brown areas and white areas. So the example here, it says a species of flower displays incomplete dominance where heterozygous individuals are a blend of the dominant and recessive colors, and so they're pink. A red flower, capital R, pollinates a white flower. Well, actually, it doesn't matter the capital or lowercase, but WW. What phenotypes will their offspring be? So sometimes you can use 2 different notations, like RR equals red, WW equals white, and then a mix of them. So if it was RW, heterozygous, it would be pink. Some other times you can just keep it simple and use our same notation where you have dominant, like this is red, equals pink. Oops, sorry, I should keep this consistent. Lowercase r equals white. And then if you had heterozygous, it would be pink. So if we were using our familiar notation, it would be like this. This is more accurate though, because red and white, not one of them is not stronger or dominant than the other. So that's why they use different letters. Okay, so in this question, you have a red flower crossed with a white flower. If you do a Punnett square, RR, WW, pretty straightforward. All of them are going to be RW as their genotype. So all offspring are going to be pink in this case. All right, so this is an example of incomplete dominance or codominance. Last type of inheritance is multiple alleles. Okay, multiple alleles, it's a type of codominance. So if you think about the prefix co, it means like together. So in this, for multiple alleles, this we usually been talking about two alleles like this or like capital A, lowercase a, but multiple alleles are more than just two. The most common example is blood types in humans. If you look at this little diagram right here, the four main blood types are type A, B, AB, and O. Let me actually make this big so you can see. So with your blood, someone who has type A blood, they have basically like markers on their blood cells, protein A. For someone who has type B, they have B proteins. Someone who has type AB blood, they have both. And then someone who has type O blood, they don't have either of those proteins, or what they call antigens. So depending on what blood type you are, you can receive blood from other blood types, but it depends. We talked about this kind of like when we're talking about immune system and just proteins on cell surfaces. If your cell doesn't recognize a protein, it attacks it. So in this case, you know, type A blood, someone who has type A, they want to get blood that they recognize. So blood that's either also type A, or they can also receive type O blood because type O doesn't have any proteins on it. So like someone who has type A blood won't reject type O. Same idea for type B blood. They want to receive blood from another person who is type B, or they can get blood from type O. Someone who has type AB blood, they're lucky because since they have both types of antigens, they can get blood from anyone. All right, their body in general, this is very basic, but they won't reject it. And then unfortunately, someone who has type O blood, they don't have any of these antigens. So that means they can only receive blood from someone else who is type O. The antigens, these represent our different alleles. So for blood types, the different alleles are A, B, and O. A, B, and O. This chart right here is a summary of the different blood types. So the phenotype is the blood type. The genotype, there's two ways of, oops, I was writing, you couldn't see. The genotype, there's two ways of writing it. One of them is using like, you use the letter I with A's and B's as the exponent, like superscript. Or you could just keep it simple. And so instead of like using the I's, you could just simplify it as AA. Now, since type O blood has no antigens, someone who has type A blood can either be homozygous, so capital A, or I don't need to do capitals and lowercase, but AA, or they could be AO. Remember, genotypes, you always have to have two letters because you get one from one parent and one from the other. Someone who has type B, their genotypes could be BB or BO. Someone who has type AB blood, their only genotype possible is AB. And then someone who has type O blood, their only genotype is OO. Okay, so both of their, like both chromosomes carry O. And then this is also a summary of what I was saying earlier of who they can receive blood from. So type O, we call type O the universal donor because they can give anyone blood. And then type AB blood, we call those people like universal recipients. They could get blood from anyone. So they're lucky. Okay, so let's do a practice problem for this. Okay, so this is what I wrote earlier. The alleles for blood type, multiple alleles. So the three alleles are A, B, and O. So the genotypes are going to be a combination of these three letters. Okay, so the question says a person with type AB blood has a child with a person who has type B blood, whose mother was type O. What are the possible blood types of their child? Okay, so the first person has type AB blood. That's easy. Okay, that's their genotype. The second person is type B. So remember how we used to, I showed you the notation. If someone is big A, or big A, little a, but we don't know. We can indicate their genotype is A dash, right? Because we don't know what the second letter is. Someone with type B blood, same thing right here. You have BB or BO. So we know they're type B, but we don't know what their second letter is yet. The clue is it says that this person, their mom was type O. So if their mom was type O, their mom had to be OO, and they could only pass on an O allele to their child. So this second person is going to be their type B, but their genotype is BO because this O came from the mom, from the mother. Okay, so these are our two parents. Do a Punnett square. A, B, B, O. This first child could have AB blood. Second one is B, B. This one is A, O. And the second one is B, O. The question says, what are the possible blood types of their child? Their child could be, so their child could have or could be basically, so you have A, they could be type A. They could be type B. These both are type B, or they could be type AB. The only one that they could not be, the child couldn't have, is type O blood. So the child could be type A, type B, or type AB. All right, so that is an example of how you would do a blood type question. Back in the day before like DNA technology was really good, the only thing that they could go off of like for crimes or like paternity would be blood types, and you kind of just do process of elimination based off of what you know. So in this case, I think one of your practice problems is like, oh, the kids at a hospital got mixed up based off the parents and based off the kids' blood types, like match up the kids. And that's how like you would use a Punnett square to do that. Okay, here's one final example. Okay, so last example, it says, a child is born with type O blood. Okay, so if someone is type O, their genotype has to be OO. The parents are type A and type B. What are each of their genotypes? Okay, so we know the child has to have OO. Now for the parents, type A, remember it could be either AA or AO. The second parent could be B, B, or BO. But if these two parents are producing a child that has type O blood like this, then that means process of elimination, they could not be homozygous, right? Because in order for the child to have O and O, they had to get those O's from someone, somewhere. And so that means that each of the parents are going to be, so the parents must be AO and BO. Okay, so those are examples of how to do each of these problems. I'm also going to post Boson videos. Well, actually, no, the Boson video you watched should have already gone over this, but I know he has other ones that are more specific to like sex-linked traits and stuff. I'm also going to, actually, if you need help, you can message me and I'll send you the key so you can check your answers. All right, but if you try to go through these problems slowly, if you need to, rewatch the video to see the examples. I think it should be enough for you to get through most of the problems, but you could always message me if you need help. All right.